this really was a culmination of a number of other publications that we put out before this. And this was really tying multiple things together. It was bringing in the genetic component. It was bringing in the strain engineering. It was bringing in years of work on cultivation, optimization, and protein purification. And it was finally just like this, like, let's just put it all together and let's just showcase, here's an example of what you could potentially do. Because so many times you put out a paper and it's just one fraction of that equation, you know? And we were trying to show, like, look, we took it all the way from construct to cultivation to purification to assay. You know, we did the whole thing in this paper, and it's like, you know, somebody else go ahead and, you know, hopefully it's useful. And, you know, it just kind of, like I said, we want to add another host into the deck of options for biotechnology and you know, production of high value products and recombinant protein.
Today we have a spotlight on a paper, and we're going to be talking about algae fermentation. How's it going, Frank?
Good, Greg. Thanks for having me.
Yeah, of course, man. Thanks. You're always welcome to come on anytime you have a paper published or any of your other papers. So
Big fan of the show. So happy to be back for my second appearance, I think.
Yeah, dude. The virus, our virus is alive, is hot. It's a hot video. It's like my third popular video. So
yeah, we'll have to revisit it soon.
So we got your paper in Algal Research right now. The title is Optimized Production of Bioactive Human Recombinant Protein from the Microalgae Chlamydomonas reinhardtii grown at high density in a fed batch bioreactor. So we have a human protein grown in algae. What was the problem that you were trying to solve there? What was the background on why you did that?
I'll start off by saying that I didn't pick the title for this paper. My co-author did.
Okay.
Yeah, this was a project that we've been working on for about 5 years, and actually even more than that. There's a lot of previous research that led up to this. What we were trying to accomplish two things at once with this paper. So we work with this model organism called Chlamydomonas. It's a green algae, and it's got a lot of cool capabilities in terms of genetic tools. You can do a lot of genetic engineering with it. It can produce recombinant proteins. So we've always had like this one half of our lab focused on like just doing genetics and trying to figure out how can we produce really unique proteins. And a lot of the times we would target mammalian proteins because we always thought it'd be cool to disrupt big pharma with a new production organism like algae versus using something like mammalian cells, which tend to be a lot more expensive to grow them. So there's the one half of it is this like strain engineering and genetics component. And then the other half of this paper is figuring out how to actually grow this in a commercially relevant manner versus just doing it all on a petri dish. Because so much research, you know, gets published on stuff that just it looks good in the petri dish, looks good in the flask, and then you try and go and scale it up and maybe grow it, and you know, doesn't work. So this paper was kind of the culmination of these two halves of the same coin here, us trying to figure them both out at the same time and actually show that we can do both the genetic engineering and some pretty sophisticated cultivation strategies using fermentation technology to get these really high titers, these really high concentrations of a bioactive recombinant protein. So that's just the overview. A lot to unpack there. So
nice. So like by recombinant protein, you mean like a gene that's been basically installed into the genome and transformed and integrated into, I'm assuming, the nuclear genome?
Yeah, exactly. So with this specific strain, we used a gene from the human genome. So we put human DNA into algae host, and the algae host produces this human protein called ICAM1 was the name of the protein, and it's a pretty popular protein. To be honest with you, I forget what we used it for therapeutically. We picked it not because of its uses, but we picked it because in order for that protein to be bioactive, it needs to have a few different post-translational modifications made to it correctly. So after your protein is first transcribed and translated, it needs to be folded, and then it needs to be glycosylated, which is when the protein is covered in sugars. And what happens when you use a mammalian cell versus like a bacterial cell versus a plant cell versus an algal cell is they do those steps all uniquely and all differently. And if you don't fold it completely correctly, and if you don't put the right sugars on it completely in the correct spots or in the correct arrangements, the protein's not active, right? So it doesn't work. And what we were able to show here is that the algae makes a bioactive protein just like a human cell would make. So at the end of this paper, we have a little assay that shows that the protein was active and it could actually be used in its natural function.
That's awesome. So we're not just talking about the ability to transcribe something or translate something or have those codons being used in a specific way, because all organisms have like a specific string of codons that they like to make for a specific amino acid, right? So they have that usage bias. Are you codon optimizing them like in order to make sure that you can get the right, I guess the right way to make the amino acid that it's making? And
yeah, of course. This is this paper is built on like 30 years of research.
Gotcha.
In this field trying to make these types of proteins, and that was like, so codon optimization is a really important step. Mm-hmm. Because different organisms will use different codons to get the same amino acids when you're making a protein. Mm-hmm. So if you optimize the genome or your construct and optimize the DNA so the codons match what your host organism is preferential to, you get better protein production.
Very cool.
That's something that's always done in a lot of organisms, and there's without going like going too far back in time and going beyond the scope of this paper. There's a lot of work that led up to this. This wasn't like a first jab. This was the 50th shot at trying to get something like this to work. But
because it's something different, right? Like than just making a fluorescent protein or something, right? Where it's not, it doesn't have a function that you can kind of like test. And I think that's like the next level that you guys looks like you were trying to achieve here.
Yeah, so the thing that's really unique about this paper is that the protein we made is secreted out of the cell.
Oh, interesting.
And when it gets secreted out of the cell, it goes through the Golgi and the ER, and that's where it gets glycosylated. And so you could make a lot of recombinant proteins in the cell. So you transform the nuclear genome, and you say, I want you to make this protein. But what you can also do is you can add this little sequence at the end, and you say, I want you to make this protein, but then I want you to send it somewhere. And you tell the cell where to send that protein. So what we would do is we could put the same protein in the same construct, but put different localization tags on the end of it. Say, make this protein, secrete it out of the cell. Make this protein, send it to the chloroplast.
Oh, that's really cool.
Protein, send it to the cytoplasm. And then we can look at how the protein looks and how bioactive it is when it comes from each of those regions. And like I said before, for this specific protein, in order for it to be bioactive, it needs to be covered in those sugars, glycosylated, just like a human cell would make it. And the only way you can get that glycosylation is when it's secreted out of the cell. So we've never, this was one of the first proteins we worked with in our lab that's actually being secreted out of the cell. Our traditional route was to make everything stay inside of the cell and take advantage of some other parts of the cell, like the chloroplast, where there's really good protein folding and protein stabilization mechanisms. So you could get like really high concentrations, but it's all stuck inside the chloroplast.
Yeah.
And, you might be able to make a lot of protein, and you might be able to make some, like antibodies. You can make a lot of those, or just like some other simple proteins that don't need to have like, all these, sugars attached to them. So we were trying to show something here, because this is what a lot of pharmaceutical companies do. This is how most protein drugs are made, is that they're secreted out of mammalian cells, and when they get secreted, they get, like I said, covered in those sugars, and then that allows them to be bioactive and attached to their receptors properly, etc. So that aspect of this was new, and we're really excited to finally show like trying to study and understand that specific pathway, that secretion pathway, and the glycosylation patterns that are associated with it, with this organism, and figure out, is it the same as a human cell, or is it the same as the bacterial cell?
Yeah, that seems incredibly important, especially with something that has like an active function, because when you're lysine or disturbing that cell and making it into like a slurry, basically, you have a chance of disrupting those proteins, right? That's like a big problem with protein extractions, is getting and stabilizing all those things, because as soon as you disrupt all those membranes, now you have things called proteases that are chewing everything up, and how do you get your product out? So it seems like this is like a good little molecular biology, as a lot of things in molecular biology are. It's something that happens naturally that we're kind of like taking and using, billions of years of evolution later. Now we're kind of, we're using it. So is the ARS1 secretion peptide, is that what you're talking about? That's actually that little thing that you're able to do. Does that make the bleomycin? That's not the actual functional thing. Are you talking about the ICAM1? So the bleomycin is the selection marker, right? That's the thing.
Right. The ARS1 is the secretion peptide.
Gotcha.
So
cool.
That's just like you can change. You can make it secretion. You can, like I said, you can send it to the cytosol. You can send it to a chloroplast.
Very cool.
You can kind of you can pull different peptide sequences and put it on there, and it'll right as it's done making the protein, it sticks that on at the end, so the cell knows, okay, just reads it like a letter address. It just says, all right, send it that way.
Is the bleomycin going with it? Is this tethered? Is this FMDV2A? Is that like tethering these two molecules together, or are they separate?
No, there's a 2A, so there's a cleaving.
Oh, okay.
It's the ICAM that has a secretion peptide and then has a flag tag on it, so we could purify it.
Wow, really cool. Very interesting. And then a flag tag, so because that allows you to use basically like a pull down method, so you could like precipitate it out of the solution really easily. Okay, wow.
Well, and that's how we do the westerns. It's more
exactly.
It's more quantitating versus
the his tag for the pull down stuff. Okay, yeah, I got you.
Yeah, we used flag resin, so you can use flag resin, which is a resin that binds to that flag peptide sequence, so you can get the protein to stick onto a resin, and you can purify it really easily. And then you can use anti-flag antibodies to image the Western blots that you have right there.
Very
so those
cool.
Westerns, they'll show you how much gross protein is in the media. So, what's really interesting when you look at that Western right there is the first couple days, you can't even image anything, right?
Mm-hmm.
There's
right here.
Titers are so low, you can't even see it. And that is an issue. This is like, I'm transitioning now to the other part of the paper here. That's the issue that we would always have, is you could do a cell transformation and you now have a new strain that's producing a product of interest, but it's making so little of it because, the promoters for this organism are still, not the best compared to like yeast. There's a lot of things that we need to still figure out in this organism. So it can't make 10% or 20% soluble protein like some other organisms can make. It makes 0.1%, right? So when you're making such small amounts of protein, it's so difficult to do assays. It's so difficult, and especially for something that you like that might have medicinal applications here, where you need to do types of like clinical trials or just like larger assays, even with like mice or cell-based assays. You still need a lot of protein to do it. And it's like, how do you make that protein? So the traditional approach was like, we would make this new cell line, and then we would grow these huge volumes of it, and then we would have to purify just like 20 liters or more, which is a lot. to get just enough protein to like show up on a Western, and that takes tons of time. It's super inefficient. It makes no sense outside of a lab. It's pretty much just proving like, is my thing even there? Is the protein that I want even there? You do all this work to figure it out. So
what's happening in these days? How is all that because I guess the bottom one is probably your control, right? The second image, is that what this is? The showing there is cells there, but it's just not, the top one is showing that it's not producing your protein of choice. So I guess what's happening in these days?
So these three graphs here. So this the top graph is the cell mass. So that's just that's the algae fraction, right?
Okay.
So that's the algae that's making the protein.
Gotcha.
And that the other half of this paper is focused on getting that as high as possible, right? Because if you have higher concentration of your host organism, you're going to have a higher concentration of the product. So that was a really difficult challenge, getting these cells to grow at high concentrations for a few different reasons.
Okay.
And then the graph below that is showing total protein.
Oh,
so that's all the protein that was in the supernatant, because there's a lot of there's cells that break open. There's normal protein secretion pathways. There's a lot of other protein that's just in the supernatant. And then the third one is showing just our target protein, right? Just
okay. Yeah. Interesting.
So there's a couple different things that are, going on there, but we're trying to show as much as possible, try to show like any good scientist, try and show all the variables so people can read it, interpret it on their own there. So.
Very cool. Very cool. So is this, I guess, wow, this is pretty long. 500 hours for the fed batch?
Yeah, it's a really, it's a long time. And it's, depends on what you compare it to. This could be seen as a very long run. I would say it's definitely more on the long side of runs when we talk about cell-based culturing.
Mm-hmm.
But for mammalian cells, they can run those for thousands of hours, pretty much until the culture dies for mammalian cells, because what they do, the strategy for producing this same type of protein in a mammalian system is you get the cell culture to a really high density, and then you put in a cell cycle inhibitor, and you force the cells to stop dividing, and then you induce their protein production pathway. So they don't produce any recombinant protein during the growth phase. They have this big growth phase, and then they just hold the culture constant, and then they run like a perfusion culture. So they'll pump media in with fresh nutrients, and then they'll pull media out that has the protein in it, right? And then you just have this cell mass that's just in a steady state, just doing nothing, just cranking this protein out into this, perfusion stream that's just going off.
What's that called? It's like a chemostat or something?
Yeah, you can call it. There's a couple different things you call it. Perfusion culture. Chemostat.
Okay.
Yeah.
Interesting. So. That's really cool. Mm-hmm. And that's what the fed batch, right? That's what the fed batch is ideas that you're continuously feeding it small amounts of media in order to keep the culture going. Yeah,
essentially the fed portion of that means that, you're adding more and more. So a batch culture would be, I added some nutrients to some water, and I grew a culture in it. And then that was it. Like the culture grew, it ate everything, it's out of food, and then it dies. That was a batch. Yeah. But in a fed batch, you continuously pump feed in, and if you don't take something out of the culture, that means your volume is going to be slowly increasing. Mm-hmm. So that's what we did in this. We didn't take anything out of the culture. We started at something like 2 liters, and we ended at 4 liters. So we pumped in 2 liters of food for this culture and allowed it to grow. So not only did it grow in density, but it also grew in total volume. So the total amount of mass inside of our fermentation vessel went up even more than is shown in that top graph there.
And what's this crash right here? The recombinant protein at the end there. Is that just it hitting the end and you're not putting in more nutrients?
Yeah, so that's where things start to get really difficult. So, I mean, if you look at that first graph, scroll back up again and look at the first graph in figure 4.
It just goes down just a little bit, but then a huge drop in the protein.
Yeah, so that little drop in biomass that's it, you're done.
Wow.
Right? So, when the culture, this is something that our goal was to hit 50 grams per liter, and we got to like 40 something, so we were close. We were trying really, really hard to get to 50, kind of arbitrary number. And it, like every time it got up there, we tried so many times, it was just like, get there. And then the next day you come in and it's like, metabolism has totally shut off, something has become limiting, right? And this is what happens in fermentation all the time is you have such a high concentration of cell mass that it just consumes everything so quickly. And if you don't have the right balance in your feed of your macronutrients and micronutrients and your carbon substrate, if everything isn't dialed in exactly, you're going to hit a wall at some point. It's going to consume everything so quickly that your limiting resource, whatever it is, it's just going to become, it'll become limited that much faster, right? Because there's such a high demand for everything. So what we did in this paper was we did something called ion-coupled mass spectrometry, which does a elemental analysis of our media. So we essentially, prior to this paper, we were only able to get to about half as concentrated. We kept hitting that wall at like 25 grams per liter, right? Kept hitting that wall there. And we're just like, what's going on? We can't figure it out. So we took our spent media after we grew a culture and sent it off for mass spec. And we got analysis back and, we figured out, okay, we're limited on, I think it was sulfur, right? So we were like, great, let's add more sulfur to the media. We adjusted our carbon to nitrogen ratios a little bit. And just by like making super minor adjustments, we got a doubling in biomass in the next run, and we're like, awesome. Like all you have to do is like figure out those little elements that you might be missing, because they make a big difference. Because like I said, the consumption rate is so high inside of these vessels. If you're missing one little thing, everything else turns off. Interesting.
So I guess like if it's crashing, I mean, what happens to make the recombinant protein? Is this is total protein though of the recombinant protein, or is this like a fraction of that? I guess like what is there recombinant protein deteriorating there? Is it actually dropping in numbers, or is it just compared to the total protein that it's lower? Like is it changing? If it's secreting,
that's a really good question. We don't have the answer to it. My educated guess would be that there is probably a large number of proteases inside the supernatant that are always there and always consuming our recombinant protein, or just the natural. I mean, like, we were running this thing at 30 degrees Celsius. The protein could just be breaking down naturally. It's a human protein, humans are about 30 degrees Celsius. So
interesting.
It should be okay, but either way, if there was something that was actively degrading that protein, that means that our production numbers are actually higher than what are represented in that bottom graph there. But as soon as you turn off production, that, consumption of the protein just takes over. So
interesting,
but more importantly, it also really shows you what's going on inside the cell. It shows you that the cells aren't making the protein anymore. They're not happy anymore. You can see the data starts to get really messy.
Right here.
At the end there, like look at that slope in the total protein and look at the slope in the biomass, right? They're going up and up and up, and then it just starts to plateau, especially in the biomass. You can start seeing that bend over. And as soon as you see that means like you're already in trouble. You let it go for another day or two and, there's no recovering it.
Yeah, it's that beautiful sigmoidal curve of microbial.
Yeah, you don't want it.
Rates.
You just want to see like exponential all the time, right?
Yeah.
But you can only go so far.
So you have your beautiful. You guys optimized the nutrients. You guys looked at your proteins, but then prove to me that it actually is functioning the way it is. Show me that you actually made a protein that it can actually do the thing that you say it can do, and that's it seems to be like this figure 6. So you guys did a binding assay, it looks like, with ICAM.
Yeah, we did a binding assay. So we used we used like traditional human ICAM versus our recombinant ICAM. Do a binding assay just to show whether or not it would actually bind to the targeted receptor, which shows like an initial activity, passes that kind of initial test. I wasn't, to be honest, I wasn't involved in that aspect of this paper. My expertise lies in the fermentation production portion of all of this.
What does ICAM do? I'm not familiar with like human
genes. Like I said before, I couldn't even remember. Like I'm not.
Yeah.
I'm not a molecular biologist.
Humans are boring anyway, man. It's okay.
Humans are boring anyway. Human protein. It says in the paper here in the beginning of it, it says ICAM is a transmembrane protein belonging to the immunoglobulin superfamily. Consists of small intracellular domains at the C-terminus, the transmembrane domain, 5 Ig-like extracellular domains in the N-terminus. The structure of this protein is characterized by glycosylation on the extracellular domains, as well as multiple loops due to disulfide bridges. So it's a pretty complex protein. And like I said, the more important part of this is the fact that we're showing you can make a human protein that looks and acts like a human protein inside of an algae cell. And I don't think this has been done before in algae, where you're actually having something secreted out of the cell like this, because this is like, we were just really trying to mimic the standard model for a lot of pharmaceutical drugs out there, which is to use mammalian cells and secrete a protein out of the cell into the supernate. We were trying to mimic that using algae. And the reason we want to do that is because it is 1,000 times cheaper to do it in a system like this. Because when you're cultivating something like a mammalian cell, the media that you have to use is extremely expensive.
Why? What part of it is expensive?
Things like hormones and just the basic media composition, using things like bovine serum, right? You need to have, I mean, you're growing a human, like you're not a human, but you're growing a mammalian cell, right? So like Chinese hamster ovaries is a very, very common one. They grow like crazy. But even so, there's human cell cultures like HeLa cells. So you have to mimic like the stuff that's going on in a mammal to get cell reproduction, right? So you have to have transferrin, you have to have different hormones, you have to have perfect temperature control. Like there's, you got to be very gentle with the cells. You can't shear because there's no cell wall on a mammalian cell, so they can break open really easily, right? So, there's a lot of challenges that come with it. And the media, just like the liquid that they grow in those cultures, costs like hundreds to thousands of dollars per liter of media.
That's
stuff.
insane.
So that's why drugs are so expensive.
Wow.
They can be really expensive to make these. So when you grow plant cells, which algae are just plant cells, it's pennies per liter for media, because you're just giving them like basic fertilizer and salt and water. They have a cell wall, so they're very robust. You could mix them really vigorously. You're not going to break them open as easily. Another cool thing is that there's no mammalian viruses in the culture. So a lot of the times what will happen with mammalian cultures is you could get like a virus. You can get a mammalian virus that just like comes into your culture and just kills all your cells, right?
Man.
And like wipes out your culture, right?
That makes sense.
And like,
dang.
Chlamydomonas, this organism, we've never been able to find a virus that attacks this species.
Mm-hmm.
We wish we could, because that's how you make really good promoters, is you use viral promoters to get like really high protein production, because viruses are really good at replicating. And that's like, it's kind of like this, it's both a pro and a con to it, because it's like, on one hand, we don't have any viruses that can kill this thing. On the other hand, we don't have the benefits of like viral genetics that we could use to increase expression. But regardless, you're not going to get a mammalian virus, that comes in and wipes out your culture. You're not going to get a mammalian virus that gets, passed through the whole purification process and can contaminate your product at the end. And it's just like overall so much cheaper. So like we're really trying to show that there's like ways you could disrupt this industry a little bit by using alternative production hosts. So that's kind of just like, that was the driving factor, really behind this paper.
That is really cool. It's so interesting too, because I mean, the fermenters and stuff on these algae fermentation runs, I mean, they're huge, right? I mean, you're working with something that can be used at its scale. That's kind of like more like the like a yeast or a bacterial fermenter, right? That you'd normally see kind of like that people would kind of picture like a beer fermentation happening, but it's algae. But you'd think that it'd be photosynthetic, but I mean, you can actually grow, Chlamydomonas and other green algae heterotrophically, or they actually are able to have and live off of carbon that's not just CO2 based from photosynthesis, which I think is like something that's really unique, right?
Yeah, exactly. And that's like, yeah, you touched on a good point there. Like, most people think algae like growing outside in a pond photosynthetically, but you can also, like we did here, you can grow them on organic carbon, like sugar and stuff, in fermentation tanks. And that's why we focused on this, this mode of production, because you get really high, like when you use fermentation, you have such a high concentration of cells. You just get really good production. You get really good product purity coming out of things. And that whole industry is so well developed. If you can develop a process and a system in a pretty much a lab scale fermentation tank, it's not too difficult to get it to scale up and work, because it kind of just becomes an issue of process engineering at that point. But if you can get fermentation to work at small scale, it's pretty easy to scale it up to go to the largest scale fermentation out there. It'll take a little extra time to figure it out, but it's so well understood, and everything that goes into fermentation in terms of the physics and the chemistry, it's so well understood and so well studied. It's kind of turnkey. If you can have like a new product, you say, great, we can do it via fermentation, and then you just go contract. You just go pay someone. You say, here's the recipe, here's the cell line, go grow it for me, please. And there's huge facilities out there where you can have these fermentation contractors just grow your product for you.
That's really cool, man. Because I heard in terms of like expensive proteins, people are, I mean, when you're talking about like, what do you want to produce in an algae and stuff, what you're looking at, like, what's the most expensive thing that we can actually produce? And I forget what year it was, but like human growth hormone grown in algae and in Chlamydomonas specifically. So I wonder like if they could use something like this secretion method to like grow something like that, or other human proteins that are currently in kind of restricted and not, these are kind of like, I mean, is this capable of being grown in yeast? Like why not just, I guess the question is like, why not just use yeast? You're using a eukaryotic organism. Why use algae versus yeast?
Yeah, that's a question we get all the time. And there's definitely ways you can do it in bacteria, you can do it in yeast, you can do it in like insect cells, you can do it in mammalian cells, tobacco leaf. Like there's a lot of different options out there when it comes to producing recombinant proteins. And I think, from what I've learned, I think the most important thing is it comes down to the specific protein that you're working with. And some, like I said, for this one, this was kind of the first time we were trying to figure out, like, it was kind of exploratory, like, can we even get something active out of an algae cell that's secreting a protein? Is the protein even going to work? Mm-hmm. But yeah, like yeast and bacteria, they're light years ahead in terms of genetics and the cultivation compared to algae, unfortunately. Yeah. I haven't worked with them enough to confidently say why you couldn't do it with them. I know that sometimes, so like with bacteria, since they're bacteria, they don't have the best protein folding machinery, being prokaryotic. They don't have the sophisticated internal cellular machinery that eukaryotes have. So you can't always produce the most complexly folded proteins from bacteria. And I've heard that with some yeasts, but there's so many yeast strains, this doesn't apply to everything. There can be issues with the glycosylation. They can hyperglycosylate, so they can put too many sugars onto proteins when they come out. So again, I think you could find a viable product, a viable protein to pick for pretty much any production system you wanted. And I think that goes for algae too. You just got to find, what's a protein or a product that is in demand that is expensive to produce, and then you just kind of go out there and say, okay, well, let's try it in bacteria. Oh, it didn't work. Let's try it in yeast. Didn't work. Great. Hey, let's try it in algae. Maybe it'll work. So it's just kind of adding to the selection of host organisms out there. I wouldn't say it is better or worse. I'm just saying it's another option, depending on, what you're trying to make. It might be the best fit.
Yeah, I think that's a great point that you made at the end, man. It's like kind of exploring all paths and keeping the paths open. And who knows, in the future, this might be the best path for some protein that we don't even know about, or that we discover that we now have this thing to go forward with, and that has a huge impact. And I think that's the coolest part about doing this type of groundbreaking science is you don't know the implications, maybe, So, and I'm sure if you had a big money protein that could make it a ton of money, that you wouldn't tell me anyway. So anyway, man, I guess
it was funny when, we did this in a 4 liter batch. Mm-hmm. And with the concentrations that we got, we back calculated it. We had about $100,000 worth of protein in 4 liters of algae culture.
Dang.
So this is the thing, you don't need a lot of these high value proteins. They are truly high value. They're extremely expensive to make. So we were sitting there, we literally had you're like, dude, if we purified all this and put it through clinical trials and got FDA approval and spent $1 billion, right? Yeah. Then we would be able to sell it. But we were just we poured it down the drain. We didn't have anything to do with it.
Yeah.
We took our samples, we collected our data, and then we were like, okay,
C'est la vie.
You did the back calculation, and we're sitting there, and we're just like, damn, that's more than both of us make in a year.
Dang.